During interphase 1, the cell is preparing to divide. The DNA is loosely organized within the nucleus as long strings. The centrosomes are close to the nucleus and close to each other. Organelles are being duplicated and the DNA is being replicated. At the end of interphase 1, one single cell has two full sets of DNA, extra organelles, and is large enough to divide. The cell is now ready to enter meiosis. In this diagram, the green and yellow chromosomes are one homologous pair, and orange and blue chromosomes are another homologous pair. During prophase 1, homologous chromosomes begin to condense and pair up. Tetrads form. A tetrad is two homologous chromosomes lined up next to each other. Since each chromosome is made up of two sister chromatids, it will look like a group of four. The nuclear membrane begins to break up and is therefore represented by a dotted line in the picture. And at the end of this phase, the nuclear membrane is completely dissolved. The centrosomes and centrioles begin to move. Spindle fibers start to assemble. The centrioles move to opposite poles of the cell. The spindle fibers are attached to the center of each chromosome, and when they move, they pull in the chromosome in a way that causes each homologous pair to line up along the equator of the cell. Each side of the cell has chromosomes from both mother and the father. The green and blue chromosomes in the diagram could be from the mother and the yellow and orange from the father. In this phase, the cell is large and slightly oval in shape. The paired homologous chromosomes separate from each other and move towards opposite sides of the cell. The sister chromatids remain attached. The spindle fibers have completed their job of pulling the homologous chromosomes away from each other and therefore disassemble. The cell goes through cytokinesis, which means that its cytoplasm is divided and two separate cells are formed. A nuclear membrane forms around the DNA of each cell. Just as in prophase 1, the centrosomes and centrioles move toward the opposite sides of the cell, and spindle fibers begin to assemble. The spindle fibers align the chromosomes along the cell's equator. Notice that in metaphase 1, we had a homologous chromosome pair lining up along the central line. One chromosome from the mother and one from the father. In metaphase 2, there is only one chromosome from each homologous pair lined up, either from the mother or from the father, because the other chromosomes from the pair is in the other cell. During anaphase 2, the sister chromatids are pulled apart from each other and move to opposite sides of the cell. In telophase 2, the two cells turn into four cells. Nuclear membranes form around the chromosomes. The spindle fibers break down. The cells go through cytokinesis again. From the one diploid cell containing 46 chromosomes that we started out with, our result is four cells called gametes or sex cells. The gametes are haploid cells, which means that they can only contain 23 chromosomes instead of 46 like the somatic cells. During meiosis 1, in metaphase 1, the homologous chromosomes randomly pair up along the equator of the cell. It is completely random if the chromosome from the mother or father that ends up facing one pole or the other in the cell. Each pair of homologous chromosomes line up independently from the other pairs. This is called random assortment and allows for 2 to the power of 23, which equals about 8 million different combinations of gametes to be produced from one human cell. During sexual reproduction, two gametes combine, and this means that 8 million times 8 million, which equals approximately 70 trillion unique babies could be born. Unless you are an identical twin, the chance that you should end up being identical to one of your siblings is therefore 1 in 70 trillion. To sum it up, a human couple can technically have 70 trillion babies with a unique genetic code. Crossing over occurs during prophase 1, meiosis 1, and allows for additional genetic variation in the offspring. During prophase 1, the homologous chromosomes begin to pair up and condense. When this takes place, crossing over occurs. Crossing over is an exchange of genetic segments between sister chromatids within a homologous pair. When the pairs line up, some chromatids are very close to each other, and segments cross. Some of these segments detach from their chromosome and reattach to the other homologous chromosome. Crossing over happens every time a germ cell divides. It can happen many times within the same pair of homologous chromosomes. When crossing over takes place, it leads to a new combination of genes and is therefore called recombination. Recombination means mixing of parental alleles. Crossing over is one type of recombination, but there are also other types of recombination events. Recombination occurs during prophase 1 and meiosis 1. There are a few reasons to why the human species and other species as well have such a great genetic variability. First of all, there is a random fertilization of gametes. Second, there is an independent assortment of chromosomes during meiosis. Third, there is also a recombination of genes, such as crossover and other recombination events. We also have mutations without further adds to the variability of the genes. During meiosis, prophase 1 crossing over commonly takes place among the sister chromatids. Sometimes errors occur during the crossing over phase. Gene duplication is one such error. Let me explain gene duplication. Sometimes the homologous chromosomes do not align properly. When crossing over occurs, some segments may differ in size. The result is that one chromosome might have double copies of a gene, and this is called duplication, while the other chromosomes may have no copy of the gene. Gene translocation is another such error. Gene translocation means that a piece of a chromosome moves to a non-homologous chromosome. Gene translocation is often reciprocal, which means that the two non-homologous chromosomes exchange gene segments with each other. Down syndrome is an example of a disease that results from the problems with meiosis. Down syndrome is the result of a non-disjunction, which is the failure of two chromosomes to separate from each other during meiosis. In the case of Down syndrome, the person ends up with three chromosomes instead of two in the 21st pair.